In part A of this lab, we're going to add 2% bromine to different samples. The first sample is heptane, an alkane. The second is hexene, an alkene, and then toluene, an aromatic compound. Then we'll compare this to an unknown compound. So first, let's focus on the alkane. In your lab manual, it says to use hexane, but we're going to use heptane instead. It should still have the same type of reaction though. So we're going to add 10 drops of this clear solution of heptane to our test tube. I didn't count them exactly because we're mostly just looking for a color change. We're not really looking for accuracy here. And then I'm going to add 3 drops of the 2% bromine solution. And you can see it's a reddish orange color. All right, so now what we're going to do is mix or agitate the solution, and then we'll see what color change occurs. So write down your observations. What color do you notice there? Later on, we'll mix other solutions together. And if you notice that it's colorless, you can just write colorless. So now we're going to move on to our second sample. So this one will be 1-hexene. So this is an alkene. So we're going to do the same thing. We're going to add 10 drops of 1-hexene to the test tube, and then we'll add 3 drops of our 2% bromine solution. Now let's agitate the test tube and see if we notice a color. If you don't see any color forming, you can write colorless. Now let's move on to our last sample. We're going to use toluene, which is an aromatic compound. So remember, toluene has a benzene ring with a methyl group attached. Again, we're going to add about 10 drops of toluene to the test tube and about 3 drops of bromine. So what color do you notice in the test tube? You can write down your observations. If you don't see a color, you can write colorless. If you do have trouble seeing some of the subtle differences between the colors, you can always ask your instructor for help. Now we're going to perform the same test on an unknown compound. So this is unknown number 2, and we're going to add 10 drops of this unknown to a new test tube, plus about 3 drops of bromine. So looking at our sample, do we see a color change or is it colorless? And can we make any conclusions about the functional group that's present in our unknown? Let's compare it to our previous tests. Which one does it match the best? Now notice the toluene and bromine color has faded over time. In part B of the lab, we're going to perform another test on those same set of samples. But this time, we're going to add a solution of potassium permanganate, KMnO4. First, we're going to add 10 drops of acetone to each test tube. This is a solvent for hydrocarbons. Then we're going to add 2 drops of our hydrocarbon sample to the same test tube, and then we'll add 2 drops of the potassium permanganate solution. So here I'm adding acetone to each test tube, but you can always go test tube by test tube. It doesn't really matter. Whatever method works best for you. And now I'm going to go through and add each of the hydrocarbons to the appropriate test tube. So we'll start with heptane, then we'll move down to 1-hexene, then toluene, and then our unknown. So now that I've added our hydrocarbon samples to each test tube, I'm now going to add 2 drops of the potassium permanganate solution to each test tube. And then we're going to record our results. So now that I've added the potassium permanganate to each sample, I'm just going to give each test tube a quick mix so that everything can react appropriately. And then we'll record any observations we have about the colors. So after about 2 minutes, we can now record our observations. So let's start with the heptane solution. What color do you observe in that test tube? You can pause the video to make your observations. Again, if you have trouble seeing the different colors, let your instructor know. So here we have 1-hexene. Here is our toluene solution, and again, you can pause the video to make any necessary observations. And our unknown. Okay, so what can we conclude about our unknown and its functional group? Here we're going to skip part C of the lab and move on to part D. We're skipping the flammability tests just for safety purposes. But in order to perform part D of the lab, we have a boiling tube and a melting point capillary. Now the melting point capillary has to fit inside of this boiling point tube, and it will be upside down. So we have to break the melting point tube. So what I'm going to do is I'm just going to make sure that this will fit, and then I'm going to wrap the melting point tube in some filter paper and just carefully break it at the point that I measured earlier. So not too difficult, although sometimes you'll get a couple shards of glass, so you do have to be careful. So now that melting point capillary will fit inside of our boiling tube. And again, it will be upside down with the open end facing the bottom of the boiling tube. So I'm going to go ahead and break the other capillaries for the other samples. And here are the other capillary tubes that I broke, and now we can set up for the boiling point test. So here I have a thermometer clamp hooked up to a ring stand, and the thermometer just fits right on the end, and then we just make sure to tighten that screw so that the thermometer stays in place. Next, we're going to take that boiling tube and put a little rubber band around it, and then we're going to attach it to the thermometer by taking the rubber band and stretching it over the thermometer. So I zoomed in so you can see the setup a little more clearly. In the next step, I'm going to add our first sample to that boiling tube. So our first sample will be heptane. Now I'm going to add one of the broken melting point tubes into the boiling tube. And again, I want to make sure that the open end of the tube is facing the bottom. Next, we're going to add 2 or 3 boiling stones to our beaker of water down below. Boiling stones just ensure that our solution doesn't boil over, so we're just going to add 2 or 3. So here you can see the boiling stones. They're quite small. They're made out of a material that doesn't react with anything that we would be using in the lab, so they're really useful. Next, we're going to lower the thermometer clamp down so that the thermometer is sitting just above the bottom of the beaker of water. Now we're going to turn on the heat on the hot plate so that the water will eventually boil. And I'll make sure to zoom in so that you can watch the experiment in action. So not much happened up until this point, so I kind of skipped the beginning part of this experiment, but you might be able to see at the very bottom of the boiling tube, there's a little bubble forming from the melting point capillary. I'll try to point at it, so hopefully you can kind of see where the bubbles are forming inside the boiling tube. So this means that we're starting to get to the boiling point of heptane, but we do need to wait for a really rapid evolution of bubbles from the melting point capillary. So at this point, you might notice the bubbles are starting to come out at a faster speed or a faster rate. So we're just going to keep an eye on that melting point capillary and those bubbles. Okay, at this point the bubbles are coming out pretty rapidly from the bottom of the melting point tube. So I'm going to turn the heat off on the hot plate. So I'll just zoom out to show you I turned it off. And now what we're going to keep an eye out for is the meniscus of the heptane within the boiling tube. So as our surrounding water cools, the heptane will also cool, and eventually it's going to be drawn up into the capillary tube. And this has to do with the vapor pressure that exists above the solution. So once we see that solution get drawn up into the capillary tube, then that is our boiling point of heptane. And here I'm starting to notice the liquid being drawn up into the melting point capillary. So you can kind of see that rising up. So this tells us that we have reached the boiling point of heptane. So I'm marking down the temperature just off screen. It's a little hard to show you the thermometer and the boiling tube at the same time, but I will give you a data table of all of the boiling point temperatures for each sample. That way you don't have to watch me do this 4 different times, but I did want to show you how it works. So now you can compare the boiling point of the unknown to the other boiling points. And now we can narrow down what our unknown compound is.